DNA is highly negatively charged. Okay, go back and look at the structure of the DNA. You will see that the DNA structure has many, because of the phosphodiester bonds, you have the phosphate groups. Right? You are familiar with this. And because of the phosphate groups, the DNA gets a negative charge. Okay, so DNA is highly negatively charged molecule. Right? It's a highly negatively charged molecule. So if you do electrophoresis, the DNA will always move from the negative pole to the positive pole. Okay, it will always move from negative pole to the positive pole. Okay, why? Because DNA is negatively charged. Okay, now, so this is something that you have to remember. This is something very, very important, and you have to remember. So this negatively charged DNA has to be packed, right? Has to be packed in a particular fashion. Okay, it has to be packed in a particular fashion so that such a big DNA can be packed into a small space within the nucleus. So this long DNA has to be packed into such a small space within the nucleus. Okay, so how is the DNA actually arranged? So if you look at the arrangement of the DNA, first of all, the DNA is wrapped around. Okay, the DNA is wrapped around something called the histone proteins. Okay, histone proteins. DNA is wrapped around the histone proteins. Okay, this is the eukaryotic DNA. You don't see histone proteins in the prokaryotes. In the bacteria, you won't see it. Okay, this is in the eukaryotic DNA. Now, DNA is wrapped around histone proteins. So I told you already that DNA is negatively charged. And these histone proteins, on the other hand, just imagine, even if they were negatively charged, then this negatively charged DNA will not wrap around this negatively charged protein. Okay, because there will be repulsion. So these histone proteins are made up of basic amino acids. Okay, made up of basic amino acids, and they have positive charge. So that because of that reason, the DNA can be wrapped around these proteins very easily. Okay, the DNA can be wrapped around these proteins very easily. So let's look at the arrangement once. Let me just take a fresh page. So let's see how these are actually arranged. Now, if you look at the arrangement, so this is the histone protein. Okay, the histone protein is made up of an octamer. When you say octamer, that means it has 8 subunits. It has 8 subunits. So if you look at the subunits, you will see there will be 2 of each. 2 of H2A, right? H2A, 2 H2As, then 2 H2Bs, 2 H2Bs, then you have 2 H3 and 2 H4s. H3 and H4. Okay, so these 8 proteins, okay, these 8 proteins will come together to form, okay, to form a barrel-like structure. This is cylindrical or barrel-like structure. Okay, to form a cylindrical or a barrel-like structure. Okay, now I told you that the DNA, and this is made up of basic amino acids. The histone is made up of basic amino acids. So since this is made up of basic amino acids, the DNA can be wrapped around this histone. The DNA can be wrapped around this histone. So this is how the DNA is actually wrapped around the histone. So 2 complete rounds. So you can see 1 round and then the second round. Okay, the DNA 2 complete rounds around the histone protein, around a single histone protein. Okay, around a single histone protein, the DNA is wrapped around twice. Okay, so if you look at the length of the DNA, okay, what's the length of the DNA that is being wrapped around? You will see that the length is around 140 to 150 base pairs. Okay, so you have the entire thing. If you look at that, it will be 100 to 140 to 150 depending on the species. Different species will have, some species will have 140, some species will have 145, some species will have 150. Okay, so first of all, this is already basic. Okay, this is already basic. So the DNA will not have any problem because of its negative charge to wrap around it, because this histone protein is positively charged. This has positive charge, DNA has negative charge, so it becomes easier for the DNA to wrap around it. That's the first thing. Second thing, in order to ensure that the wrapped DNA is not leaving the histone proteins, the wrapped DNA is not leaving the histone proteins, right? You have another protein called the H1, which is also a histone protein. This is also a histone protein. This histone protein, okay, this histone protein will act as a clamp. Okay, will act as the H1 will act as a clamp. So it's like this. So if you have the DNA like this, the protein will stay here. Okay, the protein will stay here and make sure. So it will actually press against this. It will actually press it against this. So it will make sure that the DNA is not, for any reason, the DNA is not slipping from the histone protein. Okay, it's actually making sure that it is clamped properly. It is clamped properly so that the DNA is not moving away from or slipping from the histone protein. So this complex, okay, this entire complex with 2 rounds of DNA wrapped around a histone, 2 rounds of DNA wrapped around histone is called a nucleosome. This is called a nucleosome. Okay, the entire thing is a nucleosome. Okay, now, so you have several nucleosomes formed. So let's say this is one histone and this is another histone. Okay, so you have 2 rounds of DNA here. 1, 2 rounds of DNA, right? These 2 rounds of DNA now move to the next one, then again 2 rounds of DNA, and then move to the next one. Let's say 2 rounds of DNA and move to the next one, right? So on and so forth. Okay, so that is how the DNA is wrapped around these histone proteins. Now, this part, which is not wrapped around histone proteins, but is actually linking 2 nucleosomes. It is linking, this is one nucleosome, this is one nucleosome. It's linking 2 nucleosomes. This is called the linking DNA. And this linking DNA will be around 50 to 70 base pairs. Linking DNA will be 50 to 70 base pairs. Okay, so the linking DNA will be 50 to 70 base pairs. So the entire DNA is wound around this to form a bead-like structure. You know, you have these pearl chains, right? So the chain which people actually wear, right? So just like that, you have the entire DNA. It's something like this. Okay, so your entire DNA will form a bead, like beads on a thread. So this is like a thread, and this is like, these are like beads on a thread. Okay, so let's see what happens next. Okay, now these beads on a thread, so we already saw this part. So now this part, you see this? This is one nucleosome. Okay, this part will arrange itself in the form of a rod structure. So one here, one here, one here, one here, one here. Then on the top, next layer, next layer, next layer, next layer. So the entire thing, if you look at, it will be in the shape of a rod. So the entire thing will arrange itself in the form of a rod structure. Okay, so here you can see this is partially folded one, this is tightly folded one. And the diameter of the rod structure, the diameter of this rod structure will be 30 nanometers. Will be 30 nanometers. So that's the reason this rod is called solenoid, right? This rod is called solenoid structure. And since it has 30 nanometers diameter, this is sometimes or is aptly referred to as 30 nanometer fiber. 30 nanometer fiber. So first you see the formation of nucleosomes, and these nucleosomes are arranged in a solenoid fashion. Okay, and after arranging in a solenoid fashion or a rod fashion, the diameter of the solenoid is around 30 nanometers. That's the reason we call this 30 nanometer fiber. So if you remember the structure of the proteins, we discussed primary structure, then the secondary structure. So something like that. The primary structure is where the DNA is being wrapped around the histone proteins to form beads on a thread, right? These beads on a thread are now wrapping are arranged in a fashion that forms a solenoid or a rod-like structure. The diameter of the rod-like structure is around 30 nanometers. That's the reason why it is called a 30 nanometer fiber. Okay, 30 nanometer fiber. Now it becomes easier. The 30 nanometer fiber will then arrange randomly. Okay, so you have your DNA arranged around wrapped around histone proteins. So you have 3 nucleosomes in this picture. The nucleosomes are arranged in a rod-like fashion, so one next to another in a circular fashion, okay, to form a rod-like structure. And this rod-like structure has a diameter of 30 nanometers. Okay, that's the reason why it is called a 30 nanometer fiber. Okay, so then it arranges in the form of a scaffold. Okay, this is called the chromosome scaffold. So you have a long rod now, okay, a long rod which is arranging in the form of, it is arranging randomly in this fashion, okay, random fashion. Okay, so this is called a scaffold. Okay, now these scaffolds then condense. The scaffolds, all the scaffolds will come together. Okay, you can see here, all the scaffolds are, so this is the scaffold and all the scaffolds are coming together. Okay, and then this scaffold will ultimately form the entire chromosome. That means if I take a piece of chromosome, I will see that in that piece of chromosome, the DNA is arranged in this fashion. If I zoom in further, I will see the scaffold arrangement. Then if I zoom in further, okay, if I start zooming in, I will see that this is the structure. If I zoom in further, I will see that it is wrapped around a single histone, the DNA, 2 rounds of DNA around a single histone protein. Then if I go through all that, I will see that there is DNA. Okay, so this is how the chromosomes are made. So you have DNA wrapped around histone to form nucleosomes. The nucleosomes will form a solenoid structure, which is 30 nanometers in diameter. This is called 30 nanometer fiber. The 30 nanometer fiber will arrange in scaffolds. Okay, the scaffolds finally condense to form the metaphase chromosome. Okay, to form the metaphase chromosome. Right. So this is how you will generally see chromosome as. Okay, so closer look at this chromosome will show you all this now. Now, there are a couple of things that are important. If you look at the chromosome structure, there are 2 more components which are very important. One is centromere. Okay, what is a centromere? And then what is a telomere? As the name suggests, centromere is somewhere towards the center, and telomeres are towards, telo is end. Okay, these telomeres actually comprise the ends of the DNA. Ends of the DNA. Okay, now let's look at centromere. What is centromere actually? Okay, now centromere is nothing but a region of DNA, okay, region of the chromosome, I would say, okay, region of the chromosome, okay, which is around 0.9 to 1.2 MB. Okay, so you have your DNA, so 1 KB is equal to 1000 base pairs. Okay, so next level is MB, okay, mega base pairs. Okay, so it is around, so if you take the human chromosome, if you take the chromosome 1 is around 17 MB. Chromosome 1 is around 17 MB. So the centromere region is around, okay, the region of the chromosome of this size towards somewhere towards the center. Okay, it's not always the center, we'll see. Okay, so the centromere region is around 0.2 to 1 point. So this was studied in Arabidopsis, not in human beings. This was, the centromere region is extensively studied in Arabidopsis. And in Arabidopsis, people found that the centromere region is around 0.9 to 1.2 MB. Okay, and this centromere region has many DNA repeats. Okay, repeats. What do you mean by that? So there'll be a sequence. Okay, so let's say AAG, CAG. Okay, so let's say this is a sequence AAG, CAG. Then if I see AAG, CAG, AAG, CAG, AAG, CAG, this is called repeats. Okay, this is repeating itself in the entire DNA. AAG, sorry. This is one repeat. This is another repeat. So if you take the centromere region, okay, the centromere region has 180 base pair repeats. That means this is just a 6 base pair repeat. So 1, 2, 3, 4, 5, 6 base pair repeat. If you take the centromere region of Arabidopsis has 180 base pair repeats. Okay, around 180 base pair repeats. In the human beings, you will see 171 base pair repeats. Okay, so this is just a 6 base pair repeat. 6 base pairs are repeating. So in the same way, I can't give you the entire 171 base pair sequence. Okay, but just, you don't even have to know that also. Okay, but just understand that you have 180 base pair sequence, and then the same sequence is repeating, 180 base pair sequence. Okay, how many such base pairs can be seen? The size is around 0.9 to 1.2 MB. Okay, 0.9 to 1.2 MB. Okay, so in the centromere region, you see these repeats, the repeated sequences of the DNA. So at these repeated sequences of the DNA, you see the protein called kinetochore actually binding. Okay, the kinetochore actually binds at the centromere region. So if you remember, the spindle fibers actually bind to the centromere region and pull these chromosomes apart, right? So this is your centromere region, and these are the kinetochores, the protein core that is actually binding here. And when the spindle fibers are pulling these chromosomes, the chromatids, I'm sorry, I keep saying chromosomes, these sister chromatids, right? That happens during the anaphase. Okay, during the anaphase, when these sister chromatids are being pulled apart, this in this direction, this in this direction, how are these chromatids being pulled apart? They are being pulled apart by binding to kinetochores, which are actually binding to the centromere region. Okay, which are interacting with the centromere region of the chromosome. So the kinetochore interacts with the centromere region of the chromosome, and then the spindle fibers will actually bind to the kinetochore, and these spindle fibers will actually pull the chromatids apart. Okay, and what is the centromere region? Centromere region is nothing but the DNA sequence. It has DNA sequence with 180 base pair repeats in the case of Arabidopsis, or 171 base pair repeats in the case of human beings. Okay, and those repeats actually help in binding the kinetochore. Okay, kinetochore, and this kinetochore helps in binding the spindle fibers and pulling the chromatids apart. Okay, so that is about the centromere region. That is about centromere region. Then we have telomeres. So like I said, telomeres are nothing but the ends of the DNA. Okay, so okay. Okay. So if you take your chromosome, these ends, okay, these ends highlighted in yellow color are your telomeres. Okay, so the DNA is arranged, the entire DNA molecule arranged in this chromosome, right? Towards the ends of the DNA, you always have two ends of the DNA, right? Two ends of the DNA. So one end is here and the other end is here. One end is here, the other end is here. Okay, so those ends of the DNA are called the telomeres. The ends of the DNA in a chromosome are called the ends of the DNA in a chromosome are called the telomeres. Now, if you take the telomeres also have repeated structures. Okay, a repeated DNA sequence. It also has repeated DNA sequence. Particularly, it has the sequence TTA. GGG, what is shown here, GGG ATT. I said it in the reverse direction, but same thing. Okay, GGG ATT or TTA GGG. Okay, you see this repeat sequence in the telomeres. Many repeats of this TTA, GGG, TTA, GGG. Okay, that is this is the signature sequence. So it's a 6 base pair repeat sequence. Okay, not 180 base pair or 171 base pairs in the centromere region. What you see here is a 6 base pair repeats. Okay, and this sequence is fixed in human beings. It's TTA GGG or GGG ATT. Okay, so this sequence will keep repeating many times in the telomere region. And these telomere regions, okay, these telomere regions actually interact with two types of proteins. They are called TRF1 and then TRF2. Okay, just like how your centromere region is interacting with the kinetochore, in the same way, the telomere regions are interacting with two proteins, TRF1 and TRF2. Okay, now the TRF1 actually regulates the length of the telomeres, regulates length of telomeres, and TRF2 maintains strand extensions. It maintains strand extensions. Okay, so it maintains strand extensions. So these are the two functions of the two proteins TRF1 and TRF2 which are associated with, which can be seen associated with the telomeres, which are nothing but the ends of the DNA in chromosome. And the telomere has repeated sequence, which is GGG, ATT or TTA, GGG. Okay. Now, this is in the case of human beings, humans. Okay. Now, the important thing is these telomeres will start to chip off. Okay. So, as you age, okay, as your age increases, the DNA will be damaged from the tips. So your chromosomes are actually damaged from the tips. The telomeres get destroyed. Now, how well you will respond or how well your cells will respond to this will depend on your age. Okay, so as you age, a lot of portion of this is destroyed. And if the telomeres are completely destroyed, it will start destroying the other part of the DNA. Okay, other part of the DNA. That is where you will see many problems associated with age. Okay, you see many problems associated as you age. You will see many problems. You will start facing many problems in terms of health because your telomeres are now degraded. The telomeres basically were protecting your DNA. Okay, were protecting your DNA, but now the telomeres are destroyed. As you age, the telomeres are gradually chipped off. Okay, telomeres portions are being chopped away. Okay, and once you reach a certain age, the telomeres are gone, and you have your main DNA getting destroyed, because of which the cells will lose many different functions, and because of which you will end up with health problems. Okay, so a lot of research is being done on telomeres. So how to actually ensure that the telomeres stay intact? That's the secret for staying healthy even as you age. Okay, if you can keep your telomeres, like I said, a lot of research is going on as to how these telomeres are chopped off, or how the telomeres can be stabilized better, things like that. Okay. So this is these telomeres. The role of telomeres in aging has been discovered, I think, just 5 or 6, maybe 6 to 7 years back. Somebody actually received a Nobel Prize for this. Okay, you can go back and check it out. So that is about the telomeres. Now, depending on the location of the centromere, okay, where it is located, you can have, so basically, depending on the location, the chromatids can have, so you see the length of these are long and these are short. Okay, so the short arms of a chromatid are referred to as P arms, and the long arms are referred to as Q arms. Okay, so if the centromere is more towards here, if the centromere is here, somewhere here, okay, then you have longer Q arms than the P arms. So depending on the position of the centromere, the chromosomes are of different types. Okay, the chromosomes are of different types. Okay, so if the centromere is exactly somewhere in the middle, okay, with almost equal arms, then the centromere is, this is called a metacentric chromosome. Okay, this is metacentric chromosome. Now, if the centromere is more towards one end, more towards one side, so that you see unequal arms. Okay, one smaller and a bit longer, then it is submetacentric. More towards the end with very short arms and then very long arms, then it is acrocentric. Okay, if you see the centromere towards one end without any short arms, then it becomes telocentric. So depending on the arrangement of the centromere, you can have metacentric, submetacentric, acrocentric, and telocentric type of chromosomes. Okay, so that is about